I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
With the recording started, I'll give a handful of housewarming announcements before we move on to a welcome from Tom and then hear from our presenters. So welcome to the December edition of the Cranberry Virtual Brown Bag. You should be able to see already transcripts below if you prefer to read rather than listen. If you don't see those, there should be a ... under the more and you can view transcripts. We're happy to have you have your camera on if you want to mute yourself whenever you're not talking. But as we go through the presentation today, if you have questions, you can either unmute yourself and ask a question directly. You can type a question into the chat and I'll ask it on your behalf. You can also, if you want to have a question that's asked anonymously, use the drop down and chat to Allison Jonjak and I will ask that question anonymously on your behalf. With that, I usually, click through our agenda very quickly. Today is going to be Alice Dodonder from Quebec on her fireworm timing model. Then we'll have Bruce Eklund give us an update on cranberry statistics. And finally, Gina Sideli, the new breeder from Rutgers, will be giving us an update on their program and what the future of that looks like. But I want to give Tom Lochner with the Wisconsin State Cranberry Growers Association a minute to welcome everyone and say hello.
Yeah. Well, thanks, Allison. Good afternoon, everybody, and welcome to our monthly brown bag seminar, the last one of 2023. I want to thank Allison for putting these together and also thank our speakers for today's program. Just a reminder for growers, the Wisconsin Cranberry School is coming up in January. The 24th and 25th will be at Wilderness Lodge in Wisconsin Dells. We have sent out the registration materials. There should be a link soon on our website for you to register from there. So that's what we're working on, in addition to some other things related to research and education and funding. But our next big education program will be at the Wilderness in January, and we'd like to invite everybody to be there. Just in closing, wish everybody the happiest holiday season, whatever holiday you do celebrate. We hope you have a good one, and wish you the best in the new year, and looking forward to 2024. So I'll turn it back to you, Allison.
Thanks, Tom, and thanks to Tom as well as the Growers Association for kind of co-hosting these with UW Extension over the course of the year. We're looking at long-term having some of these recorded videos chunked up and in a digestible format for people to be able to access in the future. So stay tuned for that. As far as today is concerned, all three of these presenters were at the North American Cranberry Extension and Research Workers Conference this past August, and so wanted to get to share their work with you if you were not able to make it out to that meeting. Gina had a couple of poll questions, and I might ask those to keep us interactive and primed for her presentation. Otherwise, Jennifer, if you are here, happy to pass contact over. I will go ahead and offer this first poll question that she had, which is, would you imagine that computer vision system technologies more expensive compared to traditional methods of measuring traits? Are they less expensive compared to the traditional methods of measuring traits, or are they going to be comparable? Oh. Hello, Gina, welcome. This is Gina, the new breeder for Rutgers, and I'll let you introduce yourself a little bit, and then I will show the results of we already asked the poll question on computer vision system technologies and their pricing. So go ahead and introduce yourself.
Okay, very good. Yeah, I'm Gina Sideli, and I am the new cranberry and blueberry breeder at Rutgers.
And thanks again for joining us. I know Gina was in a prior meeting that ran right up to the minute of right now, so she's our third speaker so that she can make the opportunity to meet everybody that needs to meet with her.
So I'm going to be talking about the next generation cranberries. And that is me in the cranberry bog, and I had a really good time and got some really good photos doing that. So a little bit about me. I am from the East Coast. I was born in New York on Long Island, and I moved to California to do graduate school work, my master's and my PhD and my postdoc. And definitely love California, sunny California. Very little rain, but a lot of diversity in crops. And so I've worked for 12 plus years with perennial crops. And so sunflower, there are wild perennial sunflowers. I worked with walnut and almond primarily. Walnut was my PhD and almond was my postdoc. And so that was 8 years of working with tree orchard crops. So what I wanted to begin to tell you a little bit, there's a few slides in the work that I have done, and how does that apply to what I'm going to be doing in the future. So really important for breeding is variety trials, and you probably know that because maybe you've helped participate in one, like on grower lands, and usually we have variety trials in different regions, right? So we could assess the performance and the stability. That's really what we're looking at. And so what I'm showing you here on the left is, so this is California. These are different counties, but basically this county's in the middle and this is in the north and this is in the south. And we're looking at yield. So this is pounds per acre. So that's that's a very high yield. But what these plots show, basically, after you start to analyze the data, trial data, multi-locational, so that's multi-environments, multi-years, you can better assess whether something is locally adapted. So is it adapted for New Jersey and Massachusetts, or is it adapted really for the West Coast, Wisconsin, or maybe just Oregon and Washington? And so here, nonpareil is like Stevens, right? It's like the sort of standard that's still widely planted for almonds. And you could see like it's still very high performing, but it's lower performing in Stanislaus, this county, and higher performing in this other county, Madera. So it has a better adaptation to the middle growing region. And if we were looking for a line that was like widely adapted, so very broad, we would look at like this blue one, this genotype. So there's really not much of a difference in the yields from different environments. And so we would say that this is a more stable selection that, you know, could go to market. Another way of looking at it is like, again, this is the yield, and these are the different selections and genotypes. And we would take the mean and we would look at, okay, which are the high performers, which performs above the mean? And that would help us start to look at, okay, let's look at now the quality of each of these items that perform above the mean, right? So part of my work is using computer vision systems primarily for high throughput, large scale phenotyping. And what that does in breeding is it enables us to take quantitative data on a continuous scale, So we could document like all variation. So we can have like basically sort of this bell curve of variation for each trait, versus like if we had categorical, like a 1, 2, 3, 4, you know, there's just like those bars, like bar in a bar graph, but like there's not much variation between a 1 and a 2, right? But if it was a continuous scale, you know, it'd be 1, 1.1, 1.2. So there would be more fine measurements basically. And so a way to do this is to have a camera, and it could be just a simple RGB camera, which is like from the RGB is like visual spectrum, what we see. And so it's good for discriminating color. And so for this, the growers are compensated in walnut based on the color. Yes, it's yield, but also they get paid better for lighter seed coat color, lighter pellicle color, than a dark one for walnut. And so we wanted to look at how can we better screen for color. And then downstream analysis was to look at like what genetic locations contribute to the trait. And so there's a camera and a dome with lights that are encircled, and the computer. So there's like a pull-out tray, and you take pictures of the nuts, and then use the calibration. There's a white balance to even the illumination, and then there's this color checker with true values of each color. And then we take the picture, and then there's something called segmentation, where you can see that there's numbers and there's lines around each kernel. And so we're looking to separate the kernel from the background, and then do something called color extraction. That's usually through thresholding. And then it takes these values and compares it to the algorithm takes it and compares it to those values in the color checker. So we're able to have a quantitative data for, we called it LAB scores, which was lightness, red to green and yellow to blue. And if we just did like the color scoring chart, like that would just be a 3, but you see there's a lot more variation if we have a variety of numbers. And I also use computer vision system in almonds to look at the bloom time period. And traditionally bloom time, a team would go out every few days and evaluate each tree to say, okay, this is this % blooming. So it's very subjective, and wanted a faster way to do it. And so using drones, also there was the people on the ground taking data and the drones. So the drone will fly over a orchard and take a lot of pictures. The pictures are stitched together in what's called an orthomosaic, and that orthomosaic is aligned to a Google map for its geolocation. Also, each tree is used with the Trimble unit to locate GPS location. And then there's the tree identification. So what that is you see the canopies of the trees sort of all blend into each other. Well, we picked the middle of the canopy, and we basically said, okay, this is the area we're going to look at to quantify like the % of bloom for that day. And so the algorithm will tell us basically according to the intensity of white, like the more white we have, the more bloom there was, right? And so the red is more intense bloom, so full bloom, while white or pink are less bloom. And then the images are clipped, and so you can see here is like one tree. And again, there's the segmentation, so separating the tree from the background. So it makes a binary image, and then it's thresholded. And then we just count the number of pixels in that area to say, okay, this is the % bloom for that day. So then now moving to cranberry, what I'd like to do is the breeding program has made low acid selections. So that the idea behind that was if we balance the acids, then maybe we don't have to add as much sugar, because it haven't been successful in selecting on the sugar content. There's not a lot of variation in sugar in cranberries. And so the question really is, you know, should we do a sensory panel with juice, dried fruit, fresh fruit? So that would look like having trained panelists. We would build a lexicon, which is like a dictionary to define what we're picking out in each of like the fruit, what flavor, what aroma, is it bitter, is it sweet, does it, you know, does it smell like a strawberry, or is it more grassy, like that kind of thing. And then we compare that to a standard and rate that on a scale. And then we were able to statistically analyze to say, okay, well, these selections, like these panelists like these selections because of this, and why, and what, you know, what contributes to that. So that's one of the first things that I'll be doing. Fruit rot resistance is an issue. And so along with Peter Odemans and Jeff Nahart, and then engineer Kristen Dana, we proposed, we put in a proposal. So, you know, we don't know if we got it, but we're hoping, basically to use 3 modes of data collection. And you can see in the bottom right, it would be a ground robot, a drone, and then just a fixed camera over the bog. So that would be like the human height, and then a human taking manual scoring data. And we would want to compare the quantitative data taken out of the photos, and see, you know, how different are they? Are they correlated with each other? There's already algorithms that Kristen Dana had developed that can differentiate the berries and count the berries. So it would just need some updating to discriminate and quantify the fruit rot. So fruit rot in an actual berry and even at the bog level. And then we would be looking at these 3 different levels. What is the most efficient way to take the data, whether it's through the ground robot or the drone. And then with this data, we would perform additional genetic analyses. And then the other project is false blossom disease. And we just put in a pre-proposal, again, with Jeff Nahart as the co-PD, and then Cesar, Jim, Katie at Cornell, and then Leslie and Paul from the University of Wisconsin. And this is really to understand more about, first is the genetic resistance. Can we find natural resistance in the germplasm that we have, or in some breeding lines that we have that have that early black in their background? Improving disease detection, so that would be molecular detection in the lab. And at a macro level would be, again, using these would be hyperspectral sensors to detect the disease, and using actually airplane or a satellite imagery. The pathogen and vector phenology is, little is known about the pathogen and the time it takes for the symptoms to happen. And so studying that more is something that we want to do. And then evaluating short-term management is looking at the comparison between chemical and manual removal. And then the third objective, actually, it cut off, but what I was trying to say was like a cost benefit analysis. So management suggestions, recommendations, what is going to be the best way to try to manage false blossom. And then another study that I am interested in doing with Thierry at Rutgers, the weed scientist, is evaluating some of our germplasm and some advanced breeding lines for runner capability. So this would be to hopefully replace some different herbicides that are used in those first year when the bed is being established. So can we find germplasm that has the better runner capability to fill in those gaps right away? But then, you know, as the fruit starts to develop, then sort of switch to fruit production and go from vegetative to fruit. And so not continue to runner, but then put, you know, put its resources in the fruit. And so the proof of concept would be like to take little cuttings of germplasm, and then with a camera on like a moving on tracks, to individually take pictures of plants at regular intervals to measure the growth over time. And then once we determine what maybe has the fastest rate of growth from those selections that are good yielding selections, we would bring this study outside to look at bed establishment over time. And if there is any selections that can do that better than just using you know, the traditional weed management approaches. And so I just want to say thank you for your attention, open to questions. There's my email, so feel free to contact me. I'd love to, you know, talk with you. I was just in Massachusetts. I met the many of the Massachusetts growers. So hopefully I could come sometime next year to meet some of the growers. And I really am interested from your perspective and your point of view, what research priorities you're interested in, or you feel is something that we should be looking into here in New Jersey. So thank you very much.
Thank you so much for presenting. That's a lot of good research priorities put together in a short time. I think you just joined Rutgers. Did you join formally in September?
Yeah, September 1st.
So
yeah.
We're exciting to see you hit the ground running. Had one question from the audience so far. How are the new cranberry varieties created? Is it still cross-pollination, or are you using CRISPR or something else?
Oh yeah, no, this is still traditional breeding methods. The only thing that we're doing, like I said, is using like computer vision systems to help us like better sort of with time management and better like make better selections, and then using genomics. But that's really just like screening through DNA sequencing and looking for certain variants. It's not any molecular techniques in the lab using CRISPR at this point. That potentially might be done by others, but really just for from a research standpoint.
Did you want me to go through? I already took the poll.
Oh yeah, I'm curious.
I want to go ahead and share the results on that first.
Not really like a trick question.
No,
but
just kind of like what would you expect? And so we had roughly half the people with computer vision expecting that computer vision would be more expensive, a quarter that computer vision would be less expensive, and then a quarter that the computer and traditional methods would be kind of comparable costs. So do you have
actually I don't actually have like the answer. I just wanted to see what people would say, because we didn't do like a study with an economist to see, but it's something interesting to think about, because yes, there's the initial investment, right, in the equipment, the sensors, the computer, you know, let's say paying a computer scientist to write the program for you. But once you have the pipeline, I think ultimately you're going to save time, which equals money, because you'd have to have that staff to go out and take all that data repeatedly year after year, rather than just having a computer do it for you, right? So it's not really a trick question, but you know, you're just curious to see what people thought. I think in the end you're going to save money because you're saving time, but you know.
And also retraining issues being something too, with a lot of like across a 10-year period, you might have half a dozen grad students in with different subjective rating schemes. So I think
absolutely, yeah. So there's not the consistency.
Yeah. I have one more question from the chat. What collaborations are you participating in with the other regional breeders? Are you working with Juan Zalapa? Asks
Oh yeah, yeah, definitely. Juan and I have talked to he's very friendly and very like we're very open for collaboration. Currently the collaboration is with the VATCAP, and so we're using the Rutgers germplasm, and we're both doing studies on that germplasm. But yeah, we haven't developed a project yet, but you know, it's still very early. But yes, absolutely open for collaboration with Juan.
Awesome. I'm gonna launch this second poll here, uh, guessing how many years our variety trials run for, and how long after that does it take to make a decision for cultivar release. And while we wait for people to put in some votes there, because I have hunches, but I definitely don't have the final answer. Ann Knight, if you want to unmute yourself and introduce yourself, Tom can do a formal introduction, but I see Ann here, and I'm excited to have her on with the Growers Association. Maybe Tom will do an introduction, and Ann can say
Sure. We're happy that Ann Knight has joined the Growers Association, and she's going to serve as our finance and administration manager. Right now, she's drinking out of a fire hose to absorb. Her job responsibility is also learn more about Cranberry, so we're trying to give her as much support as we can. And one of the things that we talked about was having her participate in events like this so she can learn a little bit about the industry and some of the things that some of the great things that people are doing. So I don't know, Ann, if you want to introduce yourself.
I think you did a perfect job introducing me. That's exactly what I'm doing. And it is like drinking out of a fire hose, but I'm really excited to learn everything there is to learn here.
we're glad to have you with us. A couple more questions that I'll ask while people are still answering the poll. It would be great to understand the priorities from an overall industry standpoint and from a regional perspective for breeding. And I think that kind of speaks to what your early slide with like the almond yields varying by county, as well as like what different growing conditions in different regions result in as the prime desired characteristics regionally, as well as those that are nationwide or worldwide. Now, Gina, if you have any comment to that, it might be.
Oh, so I didn't realize it was a question.
Oh, that's good. I mean, it might be, I think, a comment to future understanding research priorities. Like as you're talking to New Jersey growers, as you're talking to Massachusetts growers, hopefully you'll have the opportunity to talk to Wisconsin growers as well, and kind of sound the depth of our priorities. And if you have a desire for that, I'm happy to help coordinate.
Oh yeah, absolutely. Yeah, that would be really good to know. You know, what disease pressures. Fruit rot is really big for us down here. You know, is false blossom, you know, much of an issue? Also, like, you know, what quality characteristics? I mean, I understand like you know, you're paid basically on the yield, so that's definitely what people are most interested in. But yeah, you know, breaking that down even further.
I think there's a lot of interest lately on timing of ripeness to have our harvest logistics be spread evenly.
Yeah, that's what the Massachusetts growers said too, as well. They wanted something that was earlier. So I wouldn't guess that you would want something that's early though.
We're in good shape on the early front.
Yeah, yeah.
We're hopeful for something mid-season before
Mid-season, okay.
So we had our questions come in for the duration poll and guesses. The longest guess was 25 years.
Okay.
Shorter guess was 5 years for variety trials, and then 2 years for deciding on a cultivar. Some guessed 10 to 15 for trials and another 5 to 10 to release. One guess seems like cheating because it's from Ed Grigleski, so Ed guesses 12. Ed might have insider knowledge on that one, but
okay.
What would your answer
Yeah,
be?
I mean, I think it definitely varies, obviously, from crop to crop. We were actually 12 is a good answer. Yeah, we were doing about 12 for almonds. And I think we wanted to break it down like, because because you can't really start looking at yields for almond until year 5 or 6. Like you'll get a crop in year 4, but it doesn't really mean anything. So it's a bit longer. I think in cranberry, I wouldn't expect it to be 12, but if the bed is established, you know, in year 2, maybe it starts flowering in year 3, then we can start looking at yield in year 4, and we probably want to look at it for 5 or 6 years. So we're probably looking at about 10 years for that trial. And then, you know, just analyzing the data behind that and deciding whether there needs to be like a follow-up trial of like on a commercial setting. It kind of depends how much space was allocated for the regional variety trials. So that would be a consideration as well. And then, you know, once there's a selection, you know, then it's kind of up to like, how long does it take to propagate it, to make it, and then the patents and the licensing. So that I believe takes, you know, a few years just in and of itself. So, but then, you know, let's say we say, okay, we've made a selection, at least the growers know, okay, we have like a few years now, like before it'll be available, but you would feel empowered to be able to like look at the data to say, oh, okay, this may be something I want to invest in or not. Whether it's going to be better than, obviously we wouldn't want to release anything that's not going to be better. Like there's no point in that, but you know, if from a, you know, cost benefit standpoint, is it going to be better for the grower to change the cultivar or stick it out for a little longer and see what's coming next, like that kind of thing.
Awesome, thank you. I think that is the end of the questions that we've seen come in on the chat. I do want to thank all of our 3 speakers today for presenting, both originally back in August at NACRU, but also presenting for our Wisconsin Grower Group, as well as everyone else across the world who is able to join us today. For those who are able to attend our Cranberry School, again, the registration for Cranberry School on January 24th and 25th has gone out by email, and I think you'll be able to see that on the WSCGA website soon as well. Hope that everybody has a wonderful Christmas or holiday, and if you have any questions on anything, feel free to reach out pretty well anytime. Thank you, everybody.